Hello, I'm Dr. Mileidy Giraldo, Global Lead for Life Sciences at Lenovo HPC and AI. And today I want to talk to you about genomics analytics and how we're combining high-performance computing and AI to solve some of humanity's greatest challenges. But before I get started, let me tell you a little bit about myself. I have a background in research. Before joining Lenovo, I used to be a bioinformatics scientist at the National Center for Biotechnology Information at NIH, the National Institutes of Health. For those of you who haven't heard about NIH, this is the largest agency dedicated to biomedical research in the world. And there, I used to be one of the go-to people for anything large-scale sequence analysis, biomarker discovery, biostats, and infectious diseases and cancer. Now at Lenovo, I bridge the interaction between our HPC IT savvy experts and our biomedical customers with whom I share a background. In my team, we run the same software, the same tools that the researchers are running in the lab, but we do it with the purposes of evaluating the latest HPC technologies to test, do they help, do they hinder, are they neutral to performance? Because we really want to reduce time to scientific insight. One particular interest of ours is to create an ecosystem around population level next generation sequencing so that we can deliver the promise of precision medicine. All right, let's get started. Let me take you back to the future. So you will walk one day into your doctor's office and it will look something like this. This is no longer a place that you go to only when you're sick, but now perhaps it's called a wellness center. In this place, the healthcare that's practiced here is one that takes into account your specific genes, information about your lifestyle and your environment to treat you. This is what your physician will think of as precision health, personalized medicine. See, we want to get to a point where we can treat you in a way that we measure your individual susceptibilities to disease, where we can predict your response to specific treatments, where we know before things happen. In terms of prescribing drugs, for example, the way it happens today is we prescribe drugs based on the average person, and we may take into account your height and weight, but we want to get to a point where we take information, take into account your genetic background, your environment, and your lifestyle to do this so that we find the right drug for you, the right dosage to, again, tailor healthcare to maximize the benefit and minimize harm, to truly deliver healthcare rather than sick care. We want to move from just treating the symptoms and treating after things have happened to knowing before it happens, more predictive and proactive medicine rather than reactive. But how do we get there? Well, let me take you back to the past where everything got started. This idea of precision medicine began with sequencing the first genome. Did you know that to sequence the first genome, it took 13 years and about $3 billion to complete? And at the end, once everything was said and done, we were left underwhelmed because the scientific community had pinned all its hopes that with this one genome, we would unlock the secrets to health, longevity, be able to deliver precision medicine. But quickly, scientists realized that we needed to sequence more of us. But to get there, that was the challenge. So really, it was about mapping human variation, mapping the differences that we see and those that we can't see back to our genomes. But where are we today? Now, given that sequencing and computing costs have declined dramatically in the last 2 decades, we have gotten to a point that now genomics is everywhere. We see genomics in the lab. We see, for example, that nowadays, nearly all biomedical projects from basic research to agriculture to virology and precision health rely on high performance computing to power such complex omics analytics. Now, note that I took you away and expanded the term from genomics to omics. So in fact, it's genomics, but all the other keywords that end in omics, like transcriptomics, proteomics, metabolomics. What we found is that genomics has become so pervasive, now we can do it in such a scale that we see it everywhere. Let me give you an example with something that's front of mind to everyone today, the COVID-19 pandemic. This is how the research landscape has changed. While the mechanisms of infection of SARS-CoV-2, the virus leading to COVID-19, and The effects it has on the host immune response are largely unknown. What we know is that the highest priority efforts in the biomedical and pharmaceutical space are tackling this from multiple angles. We see research into vaccine design, identifying antiviral drug targets, um, understanding how this virus originated, trying to understand susceptibility, why some people are more susceptible than others. What all these seemingly what all these seemingly differing approaches have in common is that one of the very first steps to derive scientific insight starts with omics analytics. So genomics, transcriptomics, and proteomics is in fact in common in all of them. So we see it in the lab in many types of projects. I just gave you an example in COVID-19, but you also see it in agriculture and other fields. We also see genomics in our homes. We see it in direct to consumer sequencing. In fact, there are fields coming out today that allowed you to, take a sample of your genome and then tailor your nutrition, for example, to that. So we see it being very pervasive, so much that we also see it in the global stage, in the population level efforts. Now let me show you a map here. This is a map of the world where we're tracking some of the largest population level efforts around the world. So in green, you see here the goals stated by each of those nations or organizations as the people they want to sequence in their country. So for example, in the United States, we have the All of Us program wanting to sequence 1 million Americans and so on. So we see it all the way from the one PI lab with one sequencer to the population level efforts. But what's involved in getting my genome sequenced today? Well, it all starts with a biological sample. It could be saliva or blood. And that the sample is input into a sequencer. The sequencer transforms biological information into digital information. Now, that digital information, think of it as a puzzle. So those are just strings of letters, just like I'm showing you here. But what you get at the end of this step is just, think of it again, a bag with puzzle pieces. But the puzzle has to be put together. And it is the supercomputer, the one that does that. And that's what we call genomics analytics, is assembling the puzzle, but also comparing this puzzle, that is your genome, against another genome. So for example, we could, and in fact, we use in this step AI and machine learning, deep learning, to compare, for example, to people. If you're interested in trying to find the genetic differences between a tall person and a short person, then you would do this at this step. And you get the numerical information of that variation across those samples. Or perhaps the researcher is interested in looking at someone who's immune versus susceptible. Or think about COVID-19. You're trying to look at the genomes of people who are asymptomatic versus those who develop pneumonia and other complications. So that's what we do at this step. Now, unfortunately, this step, the part that happens in the high performance computing environment, is the largest bottleneck to date. It takes 60 to 150 hours to process one genome. That's about a week. This is not scalable. Now, if you remember in my previous slides, what we want to get to is being able to process hundreds of thousands of genomes per year. If it takes us a week for one, it's definitely not scalable. So this is the main challenge here. Now, how do we go then from the traditional healthcare, what we're practicing today, to what we want to get to, which is precision healthcare? Well, high performance computing, genomics, and AI are the gears behind this complex machinery. So, from an HPC and genomics perspective, to really make this happen, what we need to do is the following. We need to solve that problem of that bottleneck. We need to make genome processing fast. We need to increase the throughput of genomics analytics. What that means is we need to enable the researchers to not just work on one genome, but process multiple genomes at the same time and process in the order of hundreds of thousands a year. We also, to make it stick, to make it available, we need to make it affordable. We need to make it easy to use and also secure because we're dealing with human private data. All right, so those are the challenges and their requirements. Now I'm going to show you the solution that we've developed for this. We call it GHOST. It stands for Genomics Optimization and Scalability Tool. And it's a recipe that came out of original work by Intel and the Broad Institute with the GATK software. What we did is we took that original recipe, took it back to the kitchen, if you would, and improved upon it. Now we can deliver an extremely fast, high throughput, very easy to use and affordable solution that's a hardware and software bundle. Now let me explain. Ghost comes in two flavors. One that we call Ghost Base, which is optimized for cost, and also Ghost Plus, optimized for extreme performance. So what is this Ghost? What do you get with this? You get hardware and software, but it's not just hardware. We spend a lot of time in our lab benchmarking many different types of hardware, trying to figure out the hardware that really made a difference. So it's the right HPC parts, but already pre-configured specifically for omics. In addition to that, we've also done the work on the software side to pre-configure it to maximize the resources available. So it's really an interplay of the hardware and the software. We put it all in one so that the researchers don't have to spend any time doing this. And from day one, they get high performing results. Now, for those of you who are familiar with HPC, this is the recipe, this is the actual architecture. But you don't need to know about HPC to understand this. Basically, what this is a recipe for one node. One node is one computer. A supercomputer is made up of multiples of this node, or a cluster. So again, we can support all the way from the one PI lab who has just one sequencer and perhaps needs only one computer, all the way up to the population level efforts. So we can scale this out into a cluster as well. Now, let me give you some perspective. Remember when I told you that this process, when it got started, took about 13 years? And in fact, more than 13 years was only to sequence and assemble the genome. Now, there have been many more years after that actually analyzing it. As of 2019, now this same process, we, the scientific community, have brought it down to 60 to 150 hours. But given the goal that we have of delivering precision medicine, this is still a problem. Now, after we develop goals with our discoveries and goals, we are able to reduce this 13-year to 60 to 150-hour problem down to 3 hours if you use Ghost Base, or with Ghost Plus, under an hour. These are actually GPU level speeds. We are not the first to be able to reduce these, but we are the first to be able to do this with CPUs, which is a much more affordable, much more scalable type of infrastructure. So 3.3 hours with Base, under an hour with the Plus. But what it means for the researchers, which is the most important part, is that per day for each of their computers, they'll be able to process either 7 whole genomes per computer per day or 27. Okay, so that's our contribution. But now let me show you how the impact of Ghost is really staggering. And let me show you the same numbers a different way. Remember I told you that to run the typical GATK in typical environments, it takes 60 to 150 hours. Now with the solution by Intel, it got reduced down to 11 hours. When we took that solution, we brought it down to 3.3 or less than an hour. That means that we're able to deliver up to 45x or close to 200x speed up than what's available today, both in the cloud or on-premise. But what does it mean for the researchers? Well, this means that they can improve their productivity. More genomes processed means faster time to answers. And we do this with off-the-shelf components. We don't use any specialty hardware like GPUs or FPGAs. So again, this is how we keep this accessible and affordable. In addition to this, so we did this with a type of analysis that's called variant analysis. Now, we did our deep work, went deep on that one. But more importantly, what we found is that with this single architecture, you can support multiple workloads in life sciences and not just genomics. You can support and you can scale this out to support molecular dynamics, cryo-EM, and other omics workflows that you see there. And for those of you who know what that is, they are listed there for you. But more importantly, the optimizations and goals will benefit most bioinformatics software pipelines because we've optimized for workloads that require high cores, so a lot of computing power, high memory, and fast I/O. All right, so this is truly an architecture that benefits life sciences from a bioinformatics perspective. All right, so let's go back to what we talked about at the beginning. How do we really make this happen? How do we go from traditional to precision healthcare? And all right, so if HPC, genomics, and AI are the gears behind the machine, well, it's your genes, environment, and lifestyle data that become the raw material that those engines work on. Now, the work and the discoveries that we've put forth with GOES address largely genomics, HPC, and a portion of AI. Now, if you remember, we agreed that to make this happen from genomics and HPC, you would have to make genome processing fast. Well, with GOES, I've shown you that we can reach speeds now that reduce and almost eliminate that bottleneck because we deliver speeds up to 167x. We can also increase the throughput of genomics analytics. So we allow researchers to not only run more samples at the same time, but run more in hundreds of thousands of genomes per year. We're making it affordable. We're really democratizing knowledge here. So we're actually making available cutting edge technology that was available before to the few, and we're making it available to the many now. We're delivering GPU level speeds, but at CPU level cost. And we're making it very easy to use because we deliver this, again, pre-configure hardware and software. So from day one, the researchers don't have to worry about installing this very complex machinery. And again, I use machinery here as a metaphor. Right? We're also making it secure because by running GOES on premises and on hybrid systems, you're able to circumvent a lot of the problems regarding data security and data privacy that plague the public cloud. Okay, so we've done a lot of work already to enable this, to make this a reality from the genomics, HPC, and partly on the AI side. But more work needs to be done. So we need to do more work, we as a scientific community, to make this really happening, to more work on developing wearables, being able to not only make it something that is easy for people to use, but also that captures data and talks to the rest of this machinery. We need to work on improving natural language processing methods so that we can mine the electronic health records coming from the clinic. We need to really do a lot of work on methods to wrangle data, to curate, parse, mine, model, but especially cross-link data across these fields. So the data that's coming from the omics, from the wearables, be able for them to cross-link, talk to one another, and for us to mine it in a way that makes sense. Now, my call to action to society at large here is that really to make this happen, we need to do everything that we can to foster the intersection of fields and minds. Because only at the intersection of those fields, it's that we'll get the best vantage point for inspire innovation. Who should care? Who should be involved? Well, the obvious stakeholders here are, of course, the biologists, the clinicians, the healthcare providers. But to make this happen, we'll really need the help of programmers, software developers, IT HPC, mathematicians, biostatisticians, but also administrators, project managers, social workers. We'll need journalists who are informed and educating the public to make this process possible. In essence, we need you. I can't think of a better example of using technology to solve and tackle one of humanity's greatest challenges than one where a very diverse, very multicultural, very multidisciplinary team of people will have to come together to truly deliver the promise of precision medicine. It's a very exciting time to be part of this. Thank you. Now, to learn more, I'll leave you here with some collateral. Feel free to reach out on LinkedIn. We've also published a number of papers, talks, and podcasts where we've talked more about this, but feel free to reach out if you want to engage in conversation of how to make this happen. So as I mentioned in a lot of my talk, the way traditional medicine is practiced today is really based on the average person, on studies where we try to aggregate and cover as many people as possible. But really to be able to get closer to that idea of precision medicine, that's why, every time you take a drug, there's a long list of side effects. We want to avoid that from the perspective of prescribing drugs, but we also want to move away from reacting, and we want to do more on the predictive and the proactive stage. So definitely, precision medicine is really the goal of healthcare today. All these complex analytics that I've talked about would not be possible. See, no longer are researchers analyzing a single gene in a few people. We're analyzing whole genomes in multiple people. And now, with the COVID-19 pandemic, particularly, there is not only more urgency to do this faster, but there's a larger scale. Now, consider the fact that in a single milliliter of bodily fluid, there are millions to billions of different copies of the virus. Okay. Now, then consider the fact that oftentimes we want to also sample those viruses. So when we're using genomics to design a vaccine or to look for treatments, we are going to be tackling whole genomes from many viruses from many patients. So the scale here is huge. The urgency is high. So high performance computing is really the only way to tackle this, which is why we focus so much effort, so much time and resources to not only enable it, but to make it faster so that scientists can get to their discoveries faster. So think, for example, so what happens is that we take samples of the viruses that infect people. Now, the closest those viruses are to one another, then the more likely it is that they came from the same person. So here's another omics for you. We have the field of phylogenomics. And in fact, what we do is we analyze the viruses of people in a given area. So, and in fact, let me give you another omics there for you, phylogenomics. What we do is, again, we sequence the viruses, plot them on what's called a phylogenetic tree, trying to figure out how many mutations away they are from one another. So the fewer mutations, the closer they are, and then the more likely it is that they belong to the same group, came from the same parent virus in a way. And this is what's done today. It's been done all over the world to be able to track how infection is spreading. And it's exciting to see the developments that are coming out of this. People are actually trying to do this live. And genomics analytics, it's a critical aspect of being able to do this. So telemedicine is the remote monitoring of patients, so without being able to see one another. Now, while there have been many regrettable things about the pandemic, things that we're still struggling with, one of the adaptations that we all have had to come up with as a society is our familiarity with technology. This is actually something that is taking us closer to this path of precision medicine. So before, why would you have a visit with your doctor over the computer if you were used to seeing them in person? Because of this, we were forced into that path. And now we see an increased level of comfort in all levels of society, all ages, and to use the same medium that you and I are using today to communicate with one another. So that's in terms of being able to see your doctor, be more comfortable with that, be comfortable with the technology. Now, telemedicine is also used to monitor patients long term, to be able to be able to help them with adherence to methods, to taking their medicine and so on. So again, that's where the wearables will come from as well. And with the wearables will be AI in the background, HPC as well. Now, not only aggregating all the data that's coming from that, but also crunching it in the background to make it into actionable results that then we can take it back and then be able to treat that patient better and treat them in an individual basis. Well, like I mentioned in my talk, I see AI as not being a field on its own, but in fact being a methodology, being a tool that works together with high performance computing, with AI, with genomics, and perhaps other fields that we don't see at this point. So really being a methodology that's working together with other fields to crunch data, to to have data being produced, cross-linked, and turned back into actionable um, findings that will be turned into the discoveries of tomorrow, that will be turned into that actual precision medicine that we want to get to.